Good morning, everybody. Thank you for joining us. This is Benjamin Brown. Really excited that all of you guys are here to join us. Before we get started with today's meeting, I just wanted to give you guys some highlights on some things that we're excited about as we continue to move forward. Number one, our community continues to grow. We now have over 100 founding members comprising over 15 sectors, and we continue to grow rich in diversity. From a development standpoint, we are excited to get some new committees up and off the ground here in the coming weeks and month. Our community continues to grow, and we're working on some new policies as well that will further help champion that growth and development. Engagement continues to increase, not only in the community, but at industry events. We were able to be hosted at a Precision Medicine Leader Summit approximately a month ago, where we were able to engage not only with stakeholders in the community, but invited to have join some steering committee members, as well as just further talk about Stripe. From a progress standpoint, as we'll discuss now and later in the call, we're close to beginning or initiating our strategic planning process, which will seek to engage all members in our community, as well as further refining and sharpening our goals. As you may have noticed in your inbox, we released our first community newsletter this past month, which we're really excited about. And we continue to have a single-minded focus on defining what a win looks like for all of us here in the Stripe community, and enabling and advancing activities to accelerate its happening. So thanks again for joining us, and we're looking forward to today's meeting.
Okay, so we'll start with some community updates. We'll have some updates on communications, and then we have our featured presentation, CDER Perspective on Pharmacogenomics at FDA, and then we'll have a Q&A. Okay, so Stripe is a new collaborative community. We just kicked off in June, so a lot of you have gotten questions about what is Stripe going to do. Remember, Stripe is a forum to discuss and better define challenges related to pharmacogenetics testing. Generally, we found that each challenge and opportunity in the space fell into one of 10 categories. Those categories are the names of the 10 committees that we're building out to bring members with specific similar interests and experiences together and discuss specific sets of challenges. That said, we are opening applications this week for the first 3 committees under development. If you expressed interest in evidentiary standards, consensus development, or study designs through the registration form or the kickoff survey, then you will get an email with detailed instructions and a link to complete the application. The reason we are starting with these committees is to address the immediate priority for the community in developing a standardized approach to evidence evaluation for pharmacogenetics associations. Stripe is putting in place a mechanism to establish industry standards through a multi-stakeholder collaborative approach. If the 15 healthcare sectors who are represented within the community, including the FDA, can come to a consensus on what that approach would be, then we can potentially leverage those recommendations to resolve some of the most pressing concerns for the larger community. For each of the subcommittees, there are 20 seats available, and members are invited to complete an application, and there's a formal appointment process. The application will include a overview and will not only be your opportunity to share more about your interests and background, but will also be an opportunity to give us feedback on the committee objectives. If you feel you have a valuable perspective to add to these discussions and you are willing to devote the time that's required, please apply, and keep in mind that one of the cornerstones to the initiative is term limits and rotating leadership. So there will be ongoing opportunities to participate. That information will be sent out this week, and the deadline will be in early January. We'd also like to propose the need for 2 ad hoc committees. The first is a nominations committee that will help ensure adequate stakeholder group representation on each of the committees and make recommendations to the steering committee on member slates. This committee would also help identify gaps in representation and develop a recruitment plan to ensure that the larger PGX community is aware of the opportunity to participate in the Stripe collaborative community. The second ad hoc committee that we're proposing is a strategic planning committee that would develop a community-wide strategic planning survey, help review submissions, and participate in drafting the strategic plans for the community. And those plans will be open for comments to the larger member base. A link to sign up to participate on these two committees will be distributed later this week, and we'll hold an informational call in January. And just to bring it back to what Stripe is going to do, one of the most important goals for Stripe is to connect all of the different groups in PGX and serve as a platform to support and promote all the great work that's being done in this space. And our Communication Affairs Committee is essential in developing that connectedness. So I will pass it over to Tricia to talk about Stripe communications.
Hi, everyone. So I'm Tricia Kenny. I'm leading the Communication Affairs group. We've met twice as a subcommittee, and our subcommittee goals are really around 5 main areas. One, we want to, of course, build our member base. 2, we're going to work towards building out our messaging, our value proposition, and our goals, not only for the entire organization, but the different subcommittees, and put it in language that we can all share and communicate outwards. So it's clear, what we're doing and what our goals are, what our objectives are. The third goal of our group is to keep the membership informed, make sure everyone knows what we're doing, how we're doing it, who's doing it, etc. Again, to build connections and to move things forward. We're going to work towards creating engagement opportunities for all members. So, like Sara and Ben said, we had the opportunity to present at a meeting last month, get our membership involved, and get our visibility, to what we're doing here, but also what all of you are doing in your day-to-day jobs. There's going to be opportunities next year, for meetings, publications, white papers, potentially webinars, promotion of what we're doing on social, etc. So getting what we're doing out there. And then the 5th goal is around, developing critical educational tools to move, our various initiatives forward. So when the team met, I think I said earlier, but we have about 20 people signed up to participate in communications. We decided to break into 3 groups, and the groups are led. I'm leading the internal communications group. Rachel Brummer is leading the external communications group, and Sean Jones is leading the education team. So right now we're in the process of defining what those groups are working on and putting that into a plan, and we'll present that to you guys next month. Briefly, internal communications is going to focus on, developing the messaging, working with the different internals or the subcommittees, to build out value proposition messaging, etc. We are responsible for the newsletter. So if you guys have any content that you'd like, to be placed in the newsletter, please reach out to me and we'll get that going. Right now, we're aiming to put the newsletter out at least once a month, but if we have enough content, we'll put it out more. That being said, we're not going to, it won't be an every week sort of thing. So 1 to 2 a month is what we're sort of aiming towards. And then we're also working on a member directory to be defined, including, what's in that member directory and how we're going to leverage it. Because again, we want to make sure that everyone is aware of who's participating, what they're doing, and work towards networking. So Ben Kong on our group is actually leading that. So if you hear anything over the next month or so, that's why. Like I said, Rachel's leading the external group. She needs a little bit more work from us before she can really get going, but she's going to, be focused on website, social, events, articles, getting everything that we're created out into, the public view. And then Sean's leading educational effort. She has a bunch of different ideas, her and the team, thinking about different stakeholders and what the different stakeholders need. And we'll be creating, the critical content to support everything that we're doing as a collaborative community. So stay tuned next month. We'll share our detailed plans. In the meantime, if you have any questions or want to participate in any of these activities, please reach out to me or Rachel or Sean.
All right, wonderful. Thank you so much, Tricia. So without further ado, we'll move over to our featured presentation. We are very fortunate to have Robert Schuck here today from the FDA's Center for Drug Evaluation and Research. He is a clinical pharmacologist in the Division of Translational and Precision Medicine. Dr. Schuck contributes to regulatory review of investigational new drug, and biologic licensing applications to ensure that clinical pharmacology principles and precision medicine strategies are applied appropriately in all phases of drug development to help maximize benefit and reduce risk to patients. He is also involved in policy development and regulatory science projects that advance the FDA's mission to protect and promote public health. So thank you, Dr. Schuck, for joining us.
Thank you very much. 1 second, I'm trying to share my slides. Can you see my slides now?
Yep, I can see them.
Excellent. All right, so thank you very much for that introduction. I'm Bob Schuck from the Division of Translational Precision Medicine and OCP, formerly known as the Genomics and Targeted Therapy Group, and I'll be giving a CDER perspective on pharmacogenomics at FDA. I do not have any conflicts of interest. However, wanted to note that the views and opinions expressed are my own and do not necessarily represent an official FDA position. So I think within CDER, we see there's many different uses for genomics in drug development. So we've seen genomics used preemptively to validate targets for drug development, help predict toxicities that we might expect when using a drug, and define a target population. We've seen genomics used retrospectively to help explain variable responses to a drug, identify responder and non-responder populations, and identify patients that are at serious risk for drug interactions. We've seen it used prospectively in drug development to predict drug exposure, perhaps stratifying doses in a clinical study, minimize noise associated with a study, identify patients at risk for disease or an adverse event, and select patients that might respond to a drug. So there's many different uses of genomics and biomarkers that we see employed. However, when we talk specifically about pharmacogenomics, I think a lot of time we're really talking about optimizing benefits and risks of drugs. So looking at that benefit risk profile. So this slide shows, on the left-hand side, every drug has some benefits and some risks associated with its use. And we can use pharmacogenomics to really optimize this. We can avoid populations that are likely to experience adverse drug reactions. This results in a decreased risk and a better benefit risk profile. We can identify likely responders. This results in increased benefit and benefit to a greater percentage of the patients. And again, optimizes that benefit risk profile. And lastly, in the middle here, we have optimized dose. This can do either. It can either prevent adverse drug reactions that are related to higher exposures or adjust doses for patients that are going to have lower exposures in order to make sure that they're getting the proper efficacy from a drug. At the FDA, we have many different activities in the genomics field. We have drug and biomarker evaluation. So our division has a group of dedicated genomics reviewers that actually look at influencing the use of biomarkers to enhance benefit risk profile of drugs. And this can ultimately result in inclusion of pharmacogenomic information in labeling as appropriate. We have a number of different guidance and policy efforts. So we develop and implement policies related to biomarker use in drug development. We have several different partnerships and outreach programs, so this helps us engage with stakeholders and cultivate expertise on emerging issues. And we have an active regulatory science program, so we conduct pragmatic research to enhance regulatory policy and practice, and I'll dive into each of these a little bit more in the next few slides. In terms of regulatory science activities, I won't go through all of these, but over the last few years, we've had a number of different initiatives, both internally and also with external collaborators for things such as using genomics for predicting safety of drugs, looking at precision development opportunities within the drug pipeline, analyzing things like cost of biomarker development and drug development. Genetic testing patterns, safety signal strengthening. So all these different activities, and there's a very broad array, but the real goal here is to close knowledge gaps related to pharmacogenomics and its use in drug development, help us develop well-informed guidance, help expedite drug development, and help support regulatory decision making. Over the years, we've had a number of different guidance and policy efforts related to precision medicine. So this slide dates, it starts way back in 2005, where we had our initial pharmacogenomic data submissions guidance, and a co-development concept paper was also published that year. And you can see as the years go by, it gets busier and busier. 2019 has, over half a dozen different guidances that were either issued in initial draft format or gone from draft to final in that year. And it continues on. We're still very active in the guidance and policy development. It's a very rapidly evolving field. So obviously we have to keep pace with the science. One of the things that we do is very, public facing is actually including biomarkers and genetic factors in drug labeling. And this is very common these days. 427 different biomarker drug pairs were described in labeling as of June of this year. This includes 296 unique drugs and 112 different biomarkers. And these are used for a variety of different purposes in a drug label. So roughly a third are related to metabolism or transport of the drug. 41% are related to the target or pathway polymorphisms that can impact efficacy. So this is very common with the targeted therapies that are fairly commonly approved in oncology and also in some other fields as well. 26% related to immunologic or other safety reactions associated with the drug. So it's really used for a variety of different purposes. And roughly half of this is considered actionable. So 230 of these are actionable recommendations, meaning they describe a dose reduction or use with caution, something of that sort that the prescriber can take into account. Otherwise, they're descriptive of a study design or feature or presence or absence of a gene-drug interaction, so they can just be descriptive as well. And there is a systematic process that we follow in terms of determining whether or not pharmacogenomic information should be in a drug label and how that information should be used, whether or not there's recommendations versus descriptive only. So what we're really trying to get at when we're looking at this, though, is there a clinically significant gene-drug interaction? So first we're looking at the gene-drug interaction potential and the mechanism underlying it. So what fraction is metabolized by a polymorphic drug metabolizing enzyme? Are there multiple different pathways where perhaps pharmacogenic interaction is less likely to be clinically significant, or is it predominantly metabolized all by a single polymorphic drug metabolizing enzyme? Also, we can use the presence of a clinically significant drug-drug interaction that we know about to inform whether or not there might be a gene-drug interaction and whether or not we might need to request or require a study from a company. Next, we go on and we look at the human confirmation of the effect and the magnitude of the effect. So looking at the study design of the clinical data, conduct, data analysis, really looking at the clinical study design here. Also looking at the magnitude of the association. So is it a very large gene-drug interaction that results in a very substantial increase in concentrations of the drug, or is it more a smaller impact and the drug has a wide neurotherapeutic or a wide therapeutic index and it might not be as clinically significant? And lastly, we relate all this to what the clinical implications might be. So really taking into account the exposure-response relationship for the drug, both efficacy and safety. So looking at, if there's a gene-drug interaction that might result in a genetic subgroup having higher exposure to the drug, is that higher exposure really putting them into a range of concentrations where we're likely to see an adverse drug event, or is it still within the confines of where the drug can be used very safely, and by reducing the dose, we might unfortunately result in reduced efficacy for a patient? Making sure that if you're tying a actionable recommendation to a gene-drug interaction, that it's likely to improve outcomes for patients. And all this, again, impacts whether or not the information ends up in labeling, whether or not there's a recommendation versus whether it's informative, what section it ends up in labeling, whether or not a companion diagnostic might be required to be developed in order to inform the genotype. We also have a number of different other public-facing resources related to pharmacogenomics. So in February, we published our table of pharmacogenomic associations along with CDRH. This includes pharmacogenetic associations that the FDA has evaluated and believes there is sufficient evidence to support. It's not intended to affect current regulatory requirements or policies. It's really meant to be a scientific resource out there for the community. And it's limited to pharmacogenetic associations that are related to drug metabolizing enzyme or transporter genes or predisposition to adverse events. And the table is divided into 3 different tiers. So there's associations for which the data support therapeutic management recommendations, such as avoiding use or dose adjustment. There's associations for which the data indicate a potential impact on safety or response. And lastly, there's associations for which the data demonstrate a potential impact on pharmacokinetic properties only. I think it's important to note in pharmacogenomics, there's a lot of different resources out there. There's labeling, there's the table that the FDA publishes, there's CPIC guidelines, there's many different other resources that are out there. And I wanted to look at this paragraph here that was published by some members of CPIC as a letter to the editor of pharmacological research a few months ago that was in response to an article that basically describes some differences in actions that are recommended by different bodies. I really wanted to point out here, because I think this really gets at an important point. And it says, like the authors, we encourage all stakeholders in pharmacogenomics to work to improve their evidence review, communication, and transparency. I think we can all agree that that's a good idea. However, it goes on to state, PharmGKB annotations of regulatory agencies' approved drug labels, the agencies approved drug labels themselves, and CPIC pharmacogenomics guidelines each have different purposes, and efforts to harmonize them would be difficult at best. So I think it's really important to recognize what each of these resources is for and what their intent is, because different resources can provide important information, but often for different purposes, to different audiences, and in different contexts. So to look at this a little bit more deeply, prescribing information specifically is written for the healthcare practitioner, and it must contain a summary of essential scientific information that's needed for the safe and effective use of the human prescription drug or biological product. So this can mean a variety of different things. As I've already mentioned, it can be from just informational only to dose adjustments to avoiding use to recommending or requiring a test. So it can take on a very broad, different context depending on what the data are and the specific context that the drug is being used. The CPIC guidelines, on the other hand, this is directly from the CPIC website. They're designed to help clinicians understand how available genetic test results should be used to optimize drug therapy, rather than whether tests should be ordered. So they don't make recommendations on testing, whereas sometimes labeling has to do that, but they do provide recommendations on how to use that information. PharmGKB, on the other hand, is a comprehensive resource that curates knowledge about the impact of genetic variation on drug response for clinicians and researchers. So again, meant to be more of a scientific resource there. So it's really important to appreciate what each of these are for and what the target audience is. So in terms of pharmacogenomics and where we currently stand, I think we've made a lot of progress and there's a lot of opportunities going forward. So this is from the Personalized Medicine Coalition. And it looks at precision medicine approvals over the years, and it starts back in 2005, where only 5% of the drugs were considered precision medicines. As of 2014, this had jumped up to 21%, and over the last few years, it's hovered at 25 or 35% of drugs that are approved are precision medicines. So this information is being used in very meaningful ways, and I think there's still a ways to go here. It's never going to be that every drug approved is a precision drug, because that's just not necessary. A lot of drugs work for a large variety of people and don't necessarily need to be targeted towards certain genotypes, and a lot of drugs have wide therapeutic index and aren't metabolized by polymorphic drug metabolizing enzymes, so there's no need for genetic information to inform dosage. But I do think that we're missing some opportunities here. I think one of the main reasons for that is because we have a diverse set of stakeholders in pharmacogenomics. So there's healthcare providers who are interested in providing the best care they can for their patients in a minimum amount of time. There's the patients themselves, there's regulators, there's test developers, there's pharma, there's payers, want to make sure that they're getting the best value for every dollar that's spent on healthcare, and there's professional societies that help guide the medical community as well. And we have to come to a point where all these things align in order to really use pharmacogenomic information as best we can. There's a number of different barriers, I think, that we've seen to implementation of this information. Many of these are, and they come from a variety of different settings. Some of them are related to the diversity of the stakeholders that I covered on the previous slide, and some are just related to data and scientific issues. And they come from a number of different places, including the clinical care setting, where there's testing hurdles and getting the test employed. There's comfort with clinical management and alternative treatments. So people might not want to prescribers might not want to use a new drug that requires a test when they're comfortable with a different drug that they've been using for a long time. Concern about compromising efficacy. So with dose adjustments, a lot of people are very concerned if they reduce a dose based on a subgroup, genetic subgroup, that they're going to be compromising efficacy rather than preventing adverse events. In the data scientific field, there's often insufficient evidence to support certain gene-drug interactions. Specifically, there's often unclear demonstration that testing improves clinical outcomes. I think this has been a major hurdle. There's often modest genetic effects. The frequency of relevant variants can often be low, so they have to test a number of different individuals before they get something that's actually actionable, that they would have done something different than what the prescriber would have done anyways. Often the events are rare, so with rare adverse events, often these don't even show up until the post-market setting, let alone being able to attribute it to a genetic effect. All of these are big challenges with generating that data that's necessary to use pharmacogenomics in the clinic. Also from the healthcare system, there is lack of consensus in the scientific and medical community regarding evidentiary standards and how best to use pharmacogenomics, and cost effectiveness. So every time a test is done, that's at the cost of some other healthcare resource. So I think the biggest thing to push us towards moving forward and more utilizing data for pharmacogenomics is really developing that foundational data. I think it all starts in the upper left quadrant of this slide, where we have appropriate study design, conduct, and replication of results to get that reliable data that demonstrates a clinically meaningful effect for a pharmacogenomic association. Once we have those data, we can develop reliable tests that are accurate and precise, have appropriate allele coverage and consistent genotype-phenotype calls that are providing data-supported claims to clinicians and are available to everyone. After that, then you can really see where you can have a tangible benefit to pharma and payers. So when pharma and payers see lower adverse event rates and higher response rates, there's greater patient adherence there. From pharma's perspective, that means more drugs sold. From a payer's perspective, that means prevention of poor health outcomes, which again is important from their perspective. And all of this is balanced against the burden of testing, both in terms of cost and time. Lastly, once pharma and payers, I think, are on board, there's a bigger chance to generate those reliable data. In terms of healthcare provider and patients, better patient experience and better outcomes are what they want to see. And again, this has to be balanced against the cost, time, and other services that could be provided instead of testing. So again, I think this all comes back to really generating that data that demonstrates that clinically meaningful effect and makes it actionable. And then some of these other things start to fall in line after that. And there's a number of challenges to generating those data. I understand that. These come from a variety of different areas. There's testing challenges. So there's lack of standardized genotype-phenotype calls, rules for changes when new data comes available that indicates that maybe something that was considered a poor metabolizer allele is now an intermediate metabolizer allele. How do we handle dosing of drugs that are impacted by that? Allele-specific effects for certain drugs can be an issue. And inconsistent testing of different alleles can be an issue as well. In the drug development side of things, in the pre-market setting, I think there's a limited number of exposures to assess rare adverse events. As I said before, often these things aren't even known about until the post-market setting, and then once we're aware of an adverse event that's out there, we have to be able to determine if there's a genetic effect underlying that. The limited number of patients with specific metabolizer phenotypes, such as poor metabolizers, to assess PK effects in these studies. And lastly, the dose exposure response relationships are often poorly understood. This is not specific to pharmacogenomics. This is more of a drug development and clinical pharmacology issue as a whole. But often we don't have as good of an understanding of the dose exposure response relationships as we would like to. And to really utilize the information on a gene-drug interaction that increases exposure to a drug, we have to know how that's going to impact the patient's actual response to the drug. And really having better data in that area would really help us, I think. In the post-market setting, there's often data issues there as well. So the studies are often exploratory, not adequate and well controlled. There can be publication bias in terms of what information is actually made available. And adverse events are not reliably reported. And often, even if they are, there's a lack of DNA from cases. I think there's a number of opportunities for advancement in these areas. So all these kind of map back to the previous slide, but in the testing area, standardizing of genotype phenotype calls, having recommendations for essential alleles for testing, availability of authorized tests with known adequate performance, I think would really help move the field forward. In the drug development setting, best practices for drug developers, collecting DNA and assessing potential impact on genomics on PK, efficacy, and safety in all studies. This is in FDA guidance and has been for a number of years, but we don't always see it consistently done. Dedicated pharmacogenomic studies when they need to be conducted. Better fundamental understanding of exposure-response relationships for all drugs. Again, I really think that would help us utilize a lot of the pharmacogenetic information that we have. In the post-market setting, identifying associations with rare adverse events that are not possible in the pre-market setting. So linking genome data with the EMR, real-world evidence, et cetera. And I know the eMERGE network and other efforts are underway to do this. Further refining exposure-response relationship and dedicated pharmacogenomic studies to address any gaps in knowledge that remain at the time of approval, such as non-clinical and clinical studies. Even in the presence of clear data, I think we also need to know that we need an alignment of expectations. So, the FDA has taken a number of steps to assure that the tests that are out there are supported by available data. And this has been, I think, perceived at least somewhat as the FDA being unfair or unreasonable towards pharmacogenomics, but I really don't think that's the case at all. I think the FDA has actually been very progressive about using pharmacogenomics when the data support it. And there's a whole wide medical community out there that have different views on how pharmacogenomics should be used and whether or not the data currently support its use. So other groups such as American Psychiatric Society have come out and said that the evidence does not support the commercial rush of DNA tests being developed for psychiatric medications. And this is in spite of a lot of the psychiatric drug labels actually having pharmacogenomic information in them. When we added a box warning to the clopidogrel label, this is not a contemporary example, but we were accused of irrational exuberance in terms of using pharmacogenomic information. And again, this isn't recent, but the most recent AHA guidelines still do recommend against using pharmacogenomic testing for PCI. In addition, recommendations for vitamin K antagonists, the recommendation is that it may be considered. And for statin therapy, it does not recommend using pharmacogenomic testing when initiating statin therapy. So there's a wide variety of different views on where the data currently stand and what the threshold should be for implementing pharmacogenomic testing. So how do we get the reliable evidence and make it actionable? I think alignment from diverse stakeholders is important. So what is the appropriate evidence threshold? This may vary depending on the different perspective, whether you're talking about a patient or a payer or a provider. I think there's different considerations depending on the type of test that it is too. So considerations for drug metabolizing enzyme pharmacogenomics, is just exposure matching to a normal metabolizer group the level of evidence that we want to have? Or do we want to directly demonstrate differential safety efficacy relationships for specific genetic subgroups? Dose exposure response relationships and extrapolation to genetic subgroups. So can we use that dose exposure response information and align that with what we know about different genetic subgroups and come to a reasonable prediction of whether or not a subgroup might be at risk of an adverse event or lack of efficacy? Considering genetic variation that directly impacts safety or efficacy, there's things such as the frequency of the variants. Often they're very rare. Effect sizes, which can be, can vary from being very small to very large, and different alternative therapies that are available. So you don't want to contraindicate a drug when there's nothing else available, when it still has a decent benefit risk profile for the patients that are at risk. And lastly, there's a number of different ways to engage with CDER and the FDA as a whole on this. Conferences and workshops, there were over 2,300 CDER speaker requests in 2019, and we're not able to satisfy all of these, but we do a fair deal of them. So we are available to talk at conferences and workshops. Critical path innovation meetings are a setting where CDER and investigators from industry, academia, scientific consortia, patient advocacy groups, and government can all communicate to improve efficiency and success in drug development. We are also involved in a number of public-private partnerships. So these are initiatives that promote the development of new tools, methods, and approaches to foster innovation. The biomarker qualification program, this has a relatively specific context, but the mission of the BQ program is to work with external stakeholders to develop biomarkers as drug development tools. So if a pharmacogenomic test could be used as a drug development tool, it could potentially fit into this program as well. So to summarize, I really think this all starts with generating that foundational data that supports a clinically meaningful effect of gene-drug interactions and actions that can be taken to benefit patients. And then developing evidence standards among the diverse stakeholders and getting buy-in from all sorts of different stakeholders. And then we can use this information to optimize the benefit risk and maximize impact for patients. So with that, I'll stop and take any questions that anybody has.
Yeah. Robert, thank you very much. This has been here. Thanks again for that very informative presentation. We had had some questions that had been submitted by different members of the community, and we're gonna bounce around on some of these. But one of the first ones that had come up, specifically was relating to, we'd been told that CDER surveillance had identified issues with PGX test report information, and we're just curious, what specific issues have been or had been identified, and was there a specific instance of a patient harm? And if you could further elaborate on that.
What this is in regards to just in general information that has come into the agency, or?
I guess specific to pharmacogenetics test reports and test report information that's communicated in these test reports.
So, I mean, the agency comes across, information in a variety of different ways, whether it be specific adverse events that come up in, our active surveillance, or whether it be publications, or whether it be just being aware of a test that's out there that's has information that might not be supported. I'm not sure specifically what the question is getting at, but we do have a variety of different ways that we can come across different information where there's a potential safety issue with either a drug or a test associated with a drug. And once we have that information, we're obligated to act on that if we think that it's in the best interest of public health.
Okay, and there was I think a slide earlier that you talked about some of the parameters for determining how PGX information is classified on the drug label. Could you please expand a little bit further on that, on like some of the key differences between the categories, such as PGX required, PGX testing recommended, actionable, and informational?
Yeah, I mean, so, every drug that's reviewed has kind of its own caveats, and everything's its own unique situation. So, as I outlined on the one slide, we do take a systematic approach to, first of all, is there a potential for a gene drug interaction? Second of all, do we have enough data that indicate that there is in fact a real gene drug interaction? And third, how does this relate to the clinical use of the drug? So, a drug with a wide therapeutic index that, a CYP2D6 metabolizer, for instance, has, a 30% increase in exposure. That information, if it's a significant gene drug interaction, it might make it into the label in terms of being, described in section 12, which is where we have a lot of our clinical pharmacokinetic information. And the reason that it's in the label is because it's considered essential for, better understanding the clinical pharmacology of the drug. But again, if that increase in exposure, that relatively modest increase in exposure for a wide therapeutic index drug was not thought to put the patient at any risk of an adverse event, it would be very unlikely that we would recommend a dose adjustment or recommend, any type of cautionary statements or anything like that in labeling, because it would just be considered benign, much like if, hepatic impairment resulted in a small increase in exposure, but the drug, it wasn't expected to put the patient in a concentration range that would put them at risk. So all of those factors are really, is the gene drug interaction real? Do we believe it? What is the magnitude of it? And what's the clinical effect of it? And that really, the clinical effect side is, I think, where it really gets at whether or not, it's a informational only versus something stronger, such as a dose adjustment, or even as strong as being a, contraindication or something of that sort, if we really believe that patients can't safely take it if they have a certain genotype.
Another question we got quite a few submissions on is, could you, what process is in place for pharmaceutical companies to include relevant PGX information on new drug labels, and what process would drug companies undergo to add new and relevant PGX information onto existing labels?
Yeah, so in terms of the pre-market space, the company is required to conduct all studies to inform proper understanding of the clinical pharmacology of the drug. they're not necessarily required to conduct specific pharmacogenomic studies, although, as I mentioned in the presentation, we do have it in guidance that we do recommend collecting DNA in every study and conducting any analyses that are needed. So, in terms of the question is kind of being framed as if they wanted specifically to include pharmacogenomic information, the first thing would be to do an appropriate study and look at conduct the analyses appropriately and demonstrate that there's some kind of gene-drug interaction that's important for the drug. So having the data again is obviously the cornerstone of everything. In terms of incentives for doing that kind of thing, I mean it's really it's on a case-by-case basis. If a drug is predominantly metabolized by polymorphic drug metabolizing enzyme, we would certainly want to push for a study in order to better understand that. That doesn't necessarily have to be a dedicated pharmacogenetic study. It can also be part of the population PK analysis. If they're able to get enough of the various genotypes in order to have a robust analysis. In terms of the postmark, so it's really just the data that's submitted to the NDA, though. And the stronger the data are, the more likely that information is to end up in the label. In the post-market setting, there's a process by which they can submit to have the label amended. It would be similar to if they had conducted a hepatic impairment or renal impairment or drug-drug interaction study. They can submit the data from a pharmacogenomic study and any information on how they want the label updated. And that's just submitted to the application.
As I guess as a follow-up or to expand on that, earlier in your presentation, you had mentioned that from time to time, the FDA may request or require a study from a company. And I'm wondering if you could relate that to what is the FDA's current review process for existing drug labels? What does that look like?
Review process for existing labels in terms of whether or not information needs to be changed, or?
Yeah, is it like an annual thing? Is it like every 5 years? Is it quarterly, or is it just like if there's an event, I guess?
Generally, it would be event-driven. So, we have our Office of Surveillance and Epidemiology, collects all the post-marketing data that becomes available on a drug. If there was a signal there for an adverse event, for instance, that would go to the review division for assessment of whether or not that information needed to be updated. A similar process could happen for genomic information. And then, in terms of a lot of times with pharmacogenetics, it's stuff that gets published. So, our review divisions, as well as our clinical pharmacologists and our division that has, a dedicated genomics team, are all, very well aware of the literature, the guidelines that are out there, and different information that becomes available. So it can happen, organically in that way that, it becomes clear in the literature that there's an effect that's important to be in drug labeling, and we can update it in that way. Particularly, safety signals are kind of the biggest way probably that labels get updated. So once there's a safety signal out there, we can initiate a safety labeling change process in which we work with the company to update that information and have it be as clear as possible.
Yeah, thanks for that. And based on what you said and some more questions we'd been getting in from the community, our members are curious, what opportunities are there to incorporate pharmacogenetics information into the FDA's FEARS process, or the Adverse Event Reporting System?
That one I'm not entirely clear on. I'd have to talk with our colleagues in the Office of Surveillance and Epidemiology. I don't directly review those post-marketing reports that come in. So unfortunately, I can't provide a lot of clarity on that one. But if you'd give me a name to follow up with, I could look into that and get back to you.
Okay. Yeah, thanks. And a slightly different but similar question is, we've been asked, what kind of mechanisms could be created for the Stripe PGX community and others to contribute to the development of a process for regular updating of pharmacogenetics information and relevant drug labels and other FDA resources? I'm curious kind of what your take is. I know you shared a slide on, I think, engagement or on participation, but just knowing what you know about our community and what we kind of been discussing today, what kind of mechanisms may come to mind that, could work for the Stripe community?
I mean, I think the resources that are on the slide are generally meant to be, scientific discussions. There, in terms of specific mechanisms for updating labeling, those are often done in negotiation with the drug company. So I don't know necessarily that that there would be a way to engage with us. If there were a way to specifically work with the drug company where they wanted to update the labeling, that would, then they can bring that to us directly, which would be one way to do that. And then there's, for the table of pharmacogenomic associations, there is, information on how to submit information to the docket there that, if, that can inform us of different data that are out there. That's not specifically meant to be to update labeling or anything like that, but that is another way that people can submit information to the FDA on, a potentially clinically significant gene drug interaction that's out there.
A quick looping back on the FEARS database. Is that it's currently not taking any PGX information into account, or that's something they're working on?
I am not entirely sure. Again, I would have to.
Sorry.
Okay. The post-marketing safety surveillance is not something I actively participate in. So.
Okay, fair enough. And if this is the scope of something you can expand on, we get asked a lot, what is the process for determining the scope of evidence and information that can be provided to both providers and patients in pharmacogenetics reports?
So by pharmacogenomics reports, you're referring specifically to like the pharmacogenomic test label, not the drug label?
The reports that are generated with I guess, presenting the laboratory test information, the reports that are used to, I guess, decide decision making in the clinic.
I'm not as familiar with the testing regulations since that's handled by our colleagues in CDRH. I would have to follow up with them in regards of what specifically can be done. I know that for testing companies, they can submit to have meetings with CDRH in terms of, what data are needed to support specific statements.
Thank you. On one of the slides that you had, it had showed a list of, I guess, like either policies or documents that have been created from about 2005 to present. And I was curious, is there a resource or one place people could go to on the FDA's website to be able to access those, or is that like something that could be like a compilation, or where would people in the community go to kind of track all those things down?
There is a central repository where all guidances are available. It doesn't specifically, I believe, have a genomics section, but there is a free text search where you can, look through anything that had, genetic or pharmacogenetic in the title, would come up that way. In terms of, I'm not sure that there is a specific comprehensive resource that has all of those available in one spot. Our division actually used to have it on our website, but there wasn't a lot of interest in it, and we no longer do. But if there was interest in something like that, we could bring up whether or not there's a way that we could do that.
This is another, I guess, like a follow-up on something that we talked about earlier regarding the drug companies having to be the ones to take initiative to update the labels. And we're just curious, based on your experience in working with them, what if any kind of incentives come to mind on ways to either engage or further expand on ways to update the labels or include that information? What kind of incentives may exist?
I think from pharma's perspective, they really need to see that it's adding value to their drug. From my interactions with pharma, it's all, how is the testing a hurdle that's going to prevent their drug from being prescribed, or does the test, provide some kind of information to the prescriber that makes them more likely to use it? And I think that, that's why I had the slide where I really focused on, generating that data that clearly shows a, clinically meaningful gene drug interaction, and enough data that we know what to do with that information. And I really think that that's where the buy-in will stem from is, there's a lot of resistance from a pharma's perspective, I think, to have there be another test involved. They generally perceive that as something that's going to take the doctor a little bit more time, and they're going to be a little bit less likely to prescribe it. So I think they really need to see that, tangible improvement of the benefit risk profile of their drug. To put this into context, we we do work on a number of different therapeutic individualization type efforts in the Office of Clinical Pharmacology, and one I've been interested in over the last few years is using response-guided titration. So just titrating based on a biomarker for dose. And I was speaking with someone from a pharma company that was involved in development of one of the SGLT2 inhibitors for type 2 diabetes, and she mentioned during a presentation that their team was did not want more than one dose for the drug. They did not want to do any type of titration or anything like that. And I talked with her after her presentation, and it was just perceived that even just testing for HbA1c, which is done for every diabetic anyways, was perceived to be a hurdle. And that's something that they're monitoring regularly as part of clinical care for that patient anyways. So you can see where adding a pharmacogenetic test that's required before you're prescribing a drug, there could be a lot of resistance to that. So there really needs to be a demonstration of that tangible benefit of testing, and that it's really going to make their drug safer, more effective, and be used more in the population.
I think just a couple more, Robert. One we had was, in what ways has COVID-19 or the pandemic, affected CDER's priorities regarding pharmacogenetics, and what type of changes, if any, have been initiated?
COVID has obviously affected us all in a number of different ways. And I think in terms of CDER efforts, it's there have been a number of different programs that have been heavily impacted by this. Obviously, the antiretroviral groups and others have been impacted more than others. But we've really tried to keep up with all of the other activities as well. Obviously, we understand that it's not the only public health issue that's out there. Yeah, I mean, it's impacted things in a number of different ways, I think. In terms of things that are directly involved with antiretrovirals and such, our ability to, keep clinical trials going on. We've had to write a number of guidances in that space in terms of making sure that drug companies are still able to continue these clinical trials and get meaningful data in a way that's as safe as possible for all the patients. So efforts of that sort. I'm not sure specifically what else.
I just wanted to say thank you again. I know there were quite a few other questions that had come into our chat box while we were doing this that we weren't able to, that were asked, that we'll be able to send over in an email to you. But we just wanted to say thank you again for your time this morning and for speaking to our community. I know there'll be more opportunities for us to further engage, but just thank you again.
Yeah, thank you very much for the opportunity.
All right. Well, that concludes our presentation we had this morning. Wanted to put up here for you guys at a glance. Our next all members meeting is going to be on Monday, March the 8th in the new year. I know we're all excited for 2021 at 10 a.m. And also just to keep on your radars that we want to hear from you, and that we're currently initiating our community strategic planning process. You'll be hearing from us with our Pathways for Progress and PGX, both survey and planning process. So please be on the lookout for that, as well as other communications from myself and other leaders in the community. And I've just wanted to let this opportunity be, or let this be an opportunity for us to wish you and your families a very happy holidays, happy Hanukkah, Merry Christmas. We're grateful for what we've accomplished here in 2020. We're really excited about what we're going to do together in 2021. And if there are any ways the leadership of this community can be of service to you in your role at your company, please let us know. We're here to support and serve, and we just want to wish you guys all the best. So thanks again for joining us this morning. See you in the new year.